Hello, we are the Mitos, and my name is Ivana.
I'm Riker.
I'm Lorena.
And I'm David.
And I'm Lely.
She's recording, and today we are going to be comparing and contrast RNA splicing. To begin, I will be briefly describing RNA splicing and its definition. And it is said that RNA splicing is a sequence in pre-mRNA, which is corresponding the removal of introns and the connection of the exons. Also, we have self-splicing, which is requires no extra catalyzation, which means RNA is all by itself.
So in RNA splicing, we have three different mechanisms. We have group 1 intron splicing, group 2 intron splicing, and our pre-mRNA splicing. These two are fairly similar, and Riker will go into more detail how they are similar, but all of them are similar that all three cases involve the removal of the intron and then the linkage of the exon by phosphodiester bond.
Okay, so starting out, we have our group 1 introns, which kind of bind in a kind of crazy, weird shape here to your guanosines. But ultimately what happens is it gets broken off in the middle there, and it binds to the guanosine, and then your exons form together to form your RNA. Now group 2, it's a little bit different, just a little bit, because the intron actually binds to the hydroxide right there in the adenine molecule, and then it actually ultimately, whenever it binds to the adenine, will form this lariat-shaped molecule right here, and after it does that, it also forms your RNA exon.
And I'm going to go into our third one, which is pre-mRNA. And for this one, it does need a spliceosome, which is a large complex unit to help this catalyzation happen. And this right here is actually made up of 5 different subunits, and those are your SNRPs. And they have 3 major functions, and those would be to bind to the intron sequence and to recognize the intron-exon boundaries. And the second one is to hold the pre-mRNA in the correct configuration, and the third one is for the catalyzation to happen. So we do have the U1 spliceosomes that connect into the GU site, and then we have the U2 that actually connects to the branch site, which is in the middle, and then we do have the 4, 5, and 6 that bind to the intron region. And after this happens, those result in a lariat also, and that later on degrades. So this is like similar to the group 2 and how they both end in a lariat.
So in conclusion, we went over the different different splicing mechanisms.
Say in conclusion again, now that we're there.
Okay.
Yeah.
So that way I can just cut it.
Okay, let's stop.
Okay, go.
And like we're going to be right
Just
here
tell me
again.
when.
So we're going back to the
She's
splicing.
going.
Okay. In conclusion, we went over the 3 different splicing mechanisms. We went over the group 1, group 2, and the pre-mRNA, and how group 1 and group 2 are self-splicing, and where they bind to exactly, and how the phosphate bond breaks off and later connects the 2 mRNAs. The only difference with pre-mRNA is the need for the complex spliceosome to help with the actual RNA splicing.
Thank you.